Well, thank you very much. It's a great honor to be here speaking on this very special GFD summer. So I'm going to be telling you, my main specialty is stirring and mixing, but I got interested also in a lot of biological fluid problems involving microswimmers in particular that I'll talk about today. So think of my lecture as a little bit complementary to Cullum's yesterday, but except that my stirring and mixing looks nothing like his stirring and mixing.
Mhm.
And it's going to be a little bit disjointed. I apologize because there's going to be some historical reminiscences and then some attempts to talk about various aspects. So this is going to be a little bit all over the place. But let's start with paying tribute. Um, this is our good old friend Ed Spiegel, who passed away earlier this year. He was the last one of the founding fathers, and the one that I knew most, of course. He and Barbara for a while were like my second parents, and I would stay with Ed and Barbara many summers when I came here, and I will miss him dearly. I'll miss walking with him. I'm sure you guys have experienced this Ed effect where you walk with him and he's arguing a point and then suddenly he just stops to make a very strong point. He just stops walking and people would actually run into him or like have to swerve around him, but he was completely oblivious. And this obliviousness also carried to driving. We would often be driving into the cottage in the morning and he would just accelerate and decelerate kind of as he was making a point. And there were a line of cars, Woods Hole Road, of course, you can't pass anybody. So there was a long line of cars all the way to Falmouth behind Ed trying to make some important point about the genes instability or some other thing. At the time, of course, it was very frustrating, but now I look back upon it very fondly. This is Ed at George's, of course, at George Veronis as one of our annual parties there. Okay, stirring and mixing at GFD. The big summer for me was the summer of 99. As Glenn said, I was a fellow in 96. Then by 99, I was a newly minted postdoc after getting my PhD at Texas with Phil Morrison. And I was a postdoc with Alan Boozer at Columbia, who's a plasma physicist. And Alan had gotten some money to study the interface between plasma physics and mixing. I knew nothing about mixing, but I was very lucky because that very summer, Bill was lecturing. So this is Bill Young. And he gave some amazing lectures, which I think I still remember very fondly today. This was also the summer where Pascal was a fellow and Jen McKinnon and maybe others that we still keep in touch with. And these lectures really influenced me a lot in my thinking about mixing. This was the director that year. This is a picture of Neil Baumforth, who many of you know. Sorry, Neil, to embarrass you with this picture, but I just had to use it. I'm going to tell you that, the glass was not full when Neil started. So don't think ill of Nilo just based on that picture. And I want to talk a little bit about the GFD volume for that year, because I think it's an exceptional volume. Claudia Pasquero and I were the editors that year. So we worked on putting the files together and kind of taking the fellows' lectures and fellows' projects and putting all that together. But that year, Bill really, really put an enormous amount of work on these lectures, on sort of polishing them and making them look coherent. And I was a postdoc, as I said, and I even visited San Diego at the time so that we could continue editing. And these lectures contain all sorts of interesting little models of mixing for every small effect that you might think of, like anomalous dispersion, etc., and even some biological effects like this sort of anti-mixing behavior that you can get with organisms, where there's patchiness exhibited merely because of asymmetry between birth and death. As Bill would say at the time, you're born next to someone, but you die alone, which lead to an asymmetry that can create patchiness by itself without even any other effect. So one thing I love about the volume is how good Bill's writing is. So let's just quote a couple of passages, which I think are lovely. Marbled end papers and old books were produced by floating colored inks on water, stirring the surface and then capturing the swirls by carefully lowering a sheet of paper onto the inky film. This technique, probably originating in Persia in the 1400s, presses hydrodynamic correlations into the service of art. Fortunately for printers and distressingly for statisticians, a single realization does not resemble the blurry diffusion equation. So what Bill was trying to emphasize here is that most of our models are ensemble averages, meaning that you run the experiment many times with random result each time. But it's very different than looking at a single realization of a random process, which is what we're given in the real world. And this is quite a difficult difference to come to grips with. And another good passage, I have hinted darkly at problems associated with spatial filters. These issues are largely ignored by optimistic scientists. The hope is that scale separation justifies the application of diffusive closures to the coarse-grained version of a single realization. So again, the same idea that you have to be very careful when dealing with single realizations versus ensemble averages. So I really urge you to, if you're a young scientist who wants to learn about mixing, not necessarily in the traditional way of learning about parametrizations and things like that, but just learning by doing simple models. There's even some MATLAB codes in this volume. The 99 GFD volume on stirring and mixing really is, I think, one of the all-time greats, and in particular because Bill put so much work on it. Another influential summer for me on mixing, it was not about mixing really, was the 2002 summers on bounds on turbulent transport that Colin mentioned yesterday because that was the first time he came back as a lecturer. But Charlie and Fritz Bussy, I think, did the bulk of the lectures, and then there were some other lectures by Colin, Rich Kurzweil, and Peter Constantine. And in fact, it was a remarkably successful summer, I think, because bounding turned out to be quite a nice source of problems. It had the right compact mathematical formulation together with not a need for excessive amount of numerical simulations, which often, can slow down a project in the summer, that made lots of fruitful fellows that summer, fellows projects that summer. I think it was the summer that launched 1,000 bounds would be a good way of putting it. Really, a lot of papers were published that summer. But for me, it's because I started, it affected me very much because that was my first collaboration with Charlie. And we wrote our first paper on mixing with John Gibbon at Imperial. And I do take at least some minute amount of credit for I think this might be Charlie's first paper involving mixing. And he would write many papers later on, including a lot with me. And the positive aspect of bringing Charlie in to studying mixing is that he really, I think, influenced the whole crowd of applied analysts to suddenly realize this problem. And in fact, the applied analysts realized that this problem was far easier than Navier-Stokes in many ways, but still had a lot of open mysteries. And so in the last 20 years, I think the applied analysts have really, really, at least mathematically changed the game. I don't think this has yet had a big impact on the actual physics of the problem, but it has really been very important for the mathematical formulations of these problems. In particular, there's the introduction of these things called mixed norms, which I'll say a little bit about now, which has been really something that has helped a lot doing the applied analysis, meaning the rigorous mathematical analysis of mixing. But first, let me pause and bring us some memories of Woods Hole from that era. It used to be a wondrous time to be in Woods Hole in the late 90s. We would all go to the Landfall restaurant for their Monday night dance party, which were legendary. And in fact, this picture is actually from later on in the mid-2000s when Jen McKinnon revived this party. So this is us. This is Oliver Bueller. This is Pascal Garot, who you saw a few days ago. This is me with more hair. This is Nilo. And this is Marie-Louise Timmermans, who we'll see next week. So I really wish, we had these disco nights with so many different scientists that I really would love to have more pictures. In particular, I would love to have pictures of Bill dancing, but I looked through my collection. I don't think I have any. So I'm very disappointed in this, that I'm not able to embarrass more people with pictures of this. But the Landfall Monday Night Madness, as it was called, was actually canceled because it actually got a bit rowdy on Monday nights. So I think locals were not very happy about it. And now a bit of whiplash, back to mixed norms. So these were introduced by Igor Mezic's group in the mid-2000s, and Charlie and I, roughly at the same time, were looking at a similar idea. And it's this idea of measuring mixing, not by studying the variance of the concentration field, which is the typical way that it's approached, but instead by studying what's called negative Sobolev norms. So these so-called H-1 norms, or H-S in general, the norm of a concentration field. It's a kind of filtering norm in the sense that what it does to your concentration is that you integrate over the volume, but you have this inverse Laplacian operator acting on here. And the inverse Laplacian is a smoothing operator. And so this thing will decay even in the absence of diffusion if your pattern of mixing becomes complicated and filamented enough. So this norm actually captures in some sense a stirring action of a velocity field. And in fact, it's very useful from the point of view of optimization. Abstracting away the diffusivity turns out to be quite a convenient thing to do. There's a nice paper, Cullum has really written quite a few papers, and Peter Schmidt and many others have written papers where they use these negative Sobolev norms, and everybody has their favorite negative exponent here that they put in. They're basically all equivalent as long as this exponent is negative. And they turned out to be well suited to optimization. So I remember seeing a talk by Cullum on this in the cottage, where really this thing did much better than trying to optimize for lowering the variance as fast as possible. The problem with the variance, with trying to lower the variance, is that because at the beginning it doesn't really decrease with a small diffusivity, there's a constant plateau at the start, and that's basically impossible to optimize. Whereas this norm will go down immediately if you start to stir properly. It doesn't have to wait for diffusion for mixing to happen. So if you'd like to see more about this, I wrote a review several years ago on what are called multi-scale norms in that paper. The mixed norms have also helped bring in, as I said, the applied analysts. In particular, there's a group, there's a triumvirate of mathematicians, Jacob Bedrosian, Samuel Punchen-Smith, and Alex Blumenthal, who have just been on a tear recently writing these papers for a rigorous analysis of mixing in Navier-Stokes, where they prove rigorous theorems. For instance, they recently showed in 2019 that Navier-Stokes with a certain kind of random forcing will mix exponentially a passive scalar in this sense, that there's a negative Sobolev norm, so a mixed norm, which decays exponentially. And it's bounded by the so-called positive Sobolev norm. So there's something about the roughness of your initial condition that comes in. So there's no diffusion here, which is why you need to bound it in terms of this diffusion. And again, I don't think this is groundbreaking from the point of view of physics, but you should, be aware of how difficult these things actually are to prove. I think this is like a 60-page paper or something like this. And in fact, as they said, proving something in the non-random setting, however, maybe in a few hundred years, right? It's probably comparable to, the Navier-Stokes regularity problem in the sense that we just have no idea. We have no idea how to prove that deterministic dynamical systems are mixing in a rigorous sense, except in very, very simple cases. So there's interest, and that's the interesting part, right? The applied analysts or the pure mathematicians are very interested in this. So hopefully, eventually, it'll lead to some good things for applications. Okay, let's go back to the summers of GFD. So the very first, I want to say a little bit about biology at GFD, not because I'm a biologist or I'm an expert in biology, but because I don't think any of the other talks in these 2 weeks have much to do with biology. So I thought I would just review a little bit about the applications of, say, physical biology, right? The interaction of biology and fluid dynamics that have gone on during GFD. And I think the first summer where that happened was '94, when Glenn was lecturer. So I think Glenn organized that summer and was the principal lecturer. And in 2010, there was another such summer, but it was on swirling and swimming in turbulence, with Glenn again as lecturer, Antonello Provenzale, and myself as principal lecturers that summer. And I think we can all agree that that is a gorgeous picture of Antonello. So another biological summer is in 2015, where there was a summer on fluid structure interactions in living environment with Mike Shelley and Pekko Hosoi as lecturer. Of course, Pekko, I think she originally, when she was invited, she was possibly invited because she was going to talk about swimmings and slugs and things like that, and crawling, but she was into sports in those days, and she still is. And so she gave us just lectures on the hydrodynamics of sports, which were excellent. In particular, we learned, for instance, when a catcher, when there's a fly ball up in the air and you see the catcher do this little dance where they go forward and back as they run, that's because of the backspin on the ball, and the ball ends up doing a kind of ribbon trajectory, and the catcher is just tracking the ball visually. So it was a really wonderful summer for learning all these very interesting sports effects, if you want to refer to it like that. Mike referred to things that are lectured on things that were much more like there was things about microtubules and things like that. It was not a very geophysical set of lectures, but very interesting nonetheless. And in fact, just to finish summarizing the biology at GFD, so there's an annual lecture for those of you who have not necessarily attended the program there's an annual lecture called the Sears Lecture, which is funded by Mr. Sears, and it's a kind of public lecture at the GFD program. So usually there's a crowd of 100 or 200 people in the auditorium in Woods Hole, and there's really been quite a few of those. These lectures have only been going on since the early 2000s, or maybe the mid 2000s, and so quite a large number of them have been on biology. Again, physical biology, right? Of course, there's James Murray, the famous author of a textbook on mathematical biology. And then just last year, Lydia Bourguiba from MIT was lecturing on the fluid dynamics of disease transmission. I think now it'd be impossible to get Lydia to speak because she's probably has to turn down invitations from everywhere in the world now. She was working on exactly the right thing for our era. She studies sneezing and how how that makes you more likely to transmit diseases and things like that. So unfortunately, she is now, the whole world depends on research like the types of things she's doing. I say unfortunately just because it's not good that we're in a pandemic, not that I don't believe in her research, of course. Okay, so let me go back to 2010 and tell you a little bit about the types of things that I was lecturing on back in the day. So the problem that I was interested in at the time, it was actually Steele Childress who got me interested in this, and I'll return a bit more about what Steve and I did. But at the time, there was quite a lot of talk about this idea of biomixing, which is a stirring by swimming organisms. In other words, if you take a very dense concentration of organisms, like here, this is jellyfish in Papua New Guinea in the famous jellyfish lake. And this is Katia, who is a postdoc. She was a student of Dabiri and then was a postdoc at Woods Hole. She might even be a scientist at Woods Hole now. Anyways, they did experiments on jellyfish. Why don't I show you the movie? They're kind of pretty. So these are experiments where you basically study how dye is trapped around a jellyfish. So you can see there's a lot of entrainment. The dye really gets stuck for quite a while behind a jellyfish. And so I think by now that the idea that biomixing, that there could be, or biogenic mixing as it could be, as it's also called, could play a role in actually stirring the ocean. I think nobody or few people really believe that anymore. It was actually considered by Walter Munk in his famous paper, Abyssal Recipes in the 60s, but he immediately said that he didn't think it was important. And Bill Dewar was one of the people in the 2000s that revived the idea by estimating a kind of energy budget for the swimming organisms in the ocean. But I think the come down was basically the idea that, because this is not just about energy, it's also about stratification. You have to be able to do overturning, to do mixing in a stratified medium, that I think that nowadays people believe that there's, if it's important, it's a very, very small percentage of the mixing that is due to biology. Though there could be local impacts, right? Especially in things like Palau, the Palau jellyfish lake and other places. I've seen papers by limnologists, so maybe in lakes, which are not so stratified as the ocean, maybe in lakes there could be important effects. Lakes overturn more often, right? They're shallower. And so perhaps there's some effects in lakes. And also I've seen some papers involving aquaculture, because in aquaculture, you really do want to mix the medium because it's the oxygenation of the fish that comes, that is crucial, and to the density of fish that you can use in aquaculture. And so if the fish help you provide mixing, then you need to take that number into account when deciding on the parameters for your aquaculture. But today I want to talk more about the microorganisms in the lab setting, because that's one place where it's unambiguously measurable, that you can measure things about biomixing. But an aside, because I can't resist showing these slides, that there's another way that organisms can mix, and that's because that's through this process called bioturbation, which is earthworms basically going through the soil and just overturning the soil. And actually, this is the topic of Charles Darwin's last book, The Formation of Vegetable Mold Through the Action of Earthworms, with observation on their habits. And by the way, you could also buy yourself a piano at the same time. In fact, this was suggested by Josiah Wedgwood II. If you've ever heard of Wedgwood pottery, this is his son who was going to become Darwin's father-in-law. But there's a great quote, which is, "I was thus led to conclude that all the vegetable mold over the whole country has passed many times through and will again pass many times through the intestinal canals of worms." I have not looked very hard, but I've not seen models for this kinds of mixing of soil by earthworms, and it would be kind of an interesting project. I'm sure that I'm sure it has been studied. I'm not trying to say that, of course, this is a crucial thing to farming and many other things. But I'd be interested to see what kind of modeling has been done in this problem. Let's go back to biomixing, though. It's Steve Childress in around 2008, 2009 that suggested that we look at this problem by studying drift trajectories, which is also the idea that's in Katia and Dabiri's paper, Nature paper that I mentioned earlier. And it turns out this is often called Darwinian drift, the drift due to a moving body. So you drag a sphere, here this is a cylinder, you drag a cylinder through a fluid and you watch how the fluid particles move. And in fact, they move in these beautiful ribbon trajectories. In fact, this is not Darwin, this is not Charles Darwin. Well, it is Charles Darwin, but it's a different Charles Darwin. This is the grandson, Charles G. Darwin, who was at Cambridge. And but this is often referred to as Darwinian drift. And I think there's still people who confuse this with actual Charles Darwin of the evolution thing. But in fact, even though it's called Darwinian drift, these pictures are from Maxwell. So Maxwell just did this problem in quite a lot of detail in 1869, analytic formulas for these beautiful trajectories. So that's the building block, at least in an inviscid fluid. These are inviscid fluid trajectories for studying the biomixing, in the sense that we're going to think of mixing by moving or by swimming organisms as a repeated set of encounters that create these kinds of trajectories. So let me show you a numerical simulation of what I mean. This is a box. We call this the gas of swimmers, the dilute theory. So you have a box of swimmers, and they're all moving in a straight line, but randomly. And there's a little particle in the middle, and we're going to be following how that particle moves as it's kicked by the swimmers. And so you can see that the particle undergoes these loop trajectories whenever it comes, whenever a swimmer happens to come near the particle. And here I've blown up the motion of the particle, because the particle is actually doesn't move very much compared to the swimmers. But you can see that the excursions are dominated by these rather large events that have these beautiful drift trajectory structure. And the model that Steve and I made is in a way a statistical model of adding up these displacements properly based on the density of swimmers, etc. So this is our original paper in 2010, which is also when I lectured. And then later on, we wrote a paper with George Lin, who was a postdoc at the time, about the viscous case. So about the not using these drift trajectories that come from the Maxwell theory, but using actual Stokes flow solutions for the drift trajectories of a microswimmer. And in fact, that was about the same time that there were lab experiments being done in Ray Goldstein's group and Jerry Golub's group on taking microorganisms. Here, this is Chlamydomonas renarti, which is a swimming algae, and looking at how small particles, small glass or polystyrene, I don't remember exactly what the particles are made of, but small particles are placed in the fluid. Let me just show you a snapshot of the motion. This is clammy, as it's affectionately called, moving around. So you can see it has two flagella at the front. And I'm going to skip the details of the experiment, but I just want to show you the motion of the little spheres. So sorry, this is a bit too, this went a bit too far. So you can see the little spheres move around, and they can track the trajectories of all of these little spheres as the organisms move around. So they can make very detailed statistics of how fluid particles are displaced by these swimming organisms. So these were beautiful experiments that produced some very detailed results. And so, for instance, this is a graph of the probably distribution of displacements, meaning you follow a little sphere and you look how it gets displaced as a function of time by repeated encounters with the swimmers. And you plot this. So this is the size of the displacement, and the vertical is the probability of getting a displacement in that size for different number density of swimmers. So these are low volume fractions of swimmers. And you can see that as you increase the volume fraction, you broaden this distribution, as you would expect, meaning that there's more encounters with swimmers. But the solid curves are the model that I subsequently wrote down in my paper in 2015, which predicts how the number density of swimmers will affect the shape of this curve. So basically, there's only one adjustable parameters in all three of these. In the sense, there's one parameter that is a fit for all three curves. You only fit once, and then you vary the volume fraction. And you can see that the fit is wonderful all the way to even the details. There's some small deviations, but we understand the source of these deviations as well. but in particular, it's the scaling with the density that was important to get correctly. And to explain the non-Gaussianity of these distributions was also of great interest. It turns out the answer is if you wait long enough, they will eventually become Gaussian, but it takes quite a while at these low volume fractions because encounters are rather rare. In fact, I was even more gratified last year, where there's another paper by Ortlieb et al., where they redid similar experiments with leptos and all, but with an eye now to looking more precisely at trying to decide which theory fit the data better. And this is the formula from my paper in 2015 for the effective diffusivity of these little spheres. In other words, the effective diffusivity of little particles. This 0.266 is not empirical. It's actually just an integral that has to be done numerically. And then beta here is the fitting parameter that I mentioned earlier. It's the stresslet strength of a microswimmer. So it's something that can be measured or can be fitted. But you see these curves, this is the effective diffusivity coefficient. And again, this is the diffusivity of the fluid, not of the swimmers. These are two things that can be studied independently as a function of swimmer density. And you can see that all of the curves fit very nicely within experimental error in the sense that this one formula fits all these. And even the orange one here is the one where you vary the velocity of the swimmers. So, and that checks for the fact that there's this extra linearity here in the velocity of the swimmer. So it was very gratifying for me to see that this model, which is really based on very simple assumptions, but which I pushed pretty far with the statistical analysis, actually is very robust in the sense that most of the features of this experiment are explained in quite great detail by the theory from the paper. So that was very nice. Okay, let's move on to another summer. GFD 2015 was on stochastic processes. That was a great summer, and Charlie During again and Hank Dijkstra were lecturers. Stochastic processes in atmospheric and oceanic dynamics. But I have to say, I learned a lot also that summer from Oliver Buehler. And here's a great picture of Oliver Buehler that shows you that it really sucks to be tall when you're dealing with small cars. I learned a ton from the extra lectures that Oliver gave as kind of tutorials to the students, and also just from my interactions with Oliver. So I really have to thank Oliver for me learning a lot about stochastic processes that summer. In fact, it was very auspicious. I was living with Bill and Paola Chessi that summer, and we're renting Claudia's house. And our neighbor was called Ito, which I thought was amazingly auspicious. If you know anything about stochastic dynamics, you know that the Ito integral, everything is called Ito, basically. So I thought it couldn't be more auspicious that we had Ito as a neighbor that summer. I never knocked on his door and asked him whether he was related to the Ito, but I'm sure the probability is pretty close to 0. Okay, so speaking of these stochastic processes then, let me tell you a little bit about an equation that I've grown to really love called the mean exit time equation, which I think is an equation that should be used more to study transport in fluid dynamics, because it really applies even beyond the stochastic framework in the sense that it's, you don't need to actually model random processes to use this equation. So the mean exit time is the following. It's tau of x0 and t0. And tau is the expected time, so the mean time, for a fluid particle starting at this initial condition to exit the system. So I have some stirring action, and I just want to measure when a particle is going to exit. And remarkably, this satisfies a backwards advection diffusion equation. In other words, the time here, tau, as a dependent variable, as a function of t0, and grad 0 here are derivatives with respect to the not variables. There's a weird +1 source, but that's basically the clock in a way. It's like d tau, dt0 is 1. It's kind of measuring time for you. So if you just solve this equation, but you have to solve it backwards in time because it's an ill-posed equation. See, there's a negative sign forward in time. It's ill-posed because there's a negative sign in front of the ddt. So it's the adjoint equation to the advection diffusion equation. But that's not really an issue. D here is the diffusivity. And so basically it confirms something that we've also suspected, is that time is really a passive scalar. It can be advected around and you lose track of it. And when I said exits, I told you that it's the mean exit time, the mean time to reach an exit. Exits are simply determined by putting 0 Dirichlet boundary conditions on sections of the boundary that you call exits. In other words, if you have some complicated domain, you're interested when will a particle reach a certain point on those domains, you just solve the mean exit time equation with Neumann boundary conditions everywhere, but a Dirichlet condition on the exit. And the nice thing about this is that it brings in some nice tools from analysis of PDEs that can be used. And we started this kind of project in 2015. Charlie and I and Bill Young did a project with Florence Marcotte to study optimization of heat exchangers using this kind of approach. In other words, you have some fluid, and the goal is to get the particles in the fluid to hit the wall as rapidly as possible, which is what heat exchange really is doing, right? Because if a fluid particle makes it to the wall is at the cooler temperature, and so it gets cooled down. But here, this was purely a kinematic problem. We didn't put any convection or anything like this. But still, it was an instructive problem to solve to see what kind of optimal flows could be used for these kind of cooling problems. So I plan to use this equation in lots of problems in the future. And I'll come back to it a bit later in a different form. Okay. So the last part of my talk now is going to be to tell you something about a current research problem that I've been thinking about, which involves the transport and diffusion of active Brownian particles. Active Brownian particles just basically means a microswimmer or something like a microswimmer. So a microswimmer is what I showed you, right? Chlamydomonas is a microswimmer. But often these days, there's lots of active particles that are not swimmers. They're just artificial particles that you create that, because of some chemical reaction, move around in a fluid. And they move around so randomly because there's a lot of noise in the system due to thermal processes, etc. So a nice way to model such active particles is a stochastic, that this SD means a stochastic differential equation of this form. So you have a rate of change of your x coordinate, your y coordinate, and the angle of swimming. This is going to be a two-dimensional model. So you write that basically all three of them have a Brownian motion associated with them. So dW1, dW2, dW3 are what's called a Brownian motion, or a noise affecting the system, with a diffusivity dx, dy, and d theta. But the x direction has an extra propulsion. And this is the swimming part. This is saying that the particle actually likes to move along its capital X axis. So these equations are formulated in the frame of the swimmer. So capital X here is some distinguished axis of swimming, and capital Y is perpendicular to that axis. And the noises are independent in those axes. Notice that d theta here has units of inverse time, unlike spatial diffusion, which is length squared over time. You'll see that later when we make dimensionless numbers. And you transform these equations into lab frame coordinates. So, you don't want to solve these in the frame of the swimmer. You want to solve this into little x, little y, and theta, which are in the lab frame of the swimmer. And because we didn't set dx equal to dy, this is anisotropic diffusivity, you get this anisotropy in the evolution. And so this is actually the stochastic differential equation that we want to solve, this coupled differential equation. One way to examine such equations is to look at what's called let me show you some sample paths first. So what do the paths look like? In fact, this equation is very different from Brownian motion in that the paths have a certain degree of smoothness because of the UDT term. So you can say that a swimmer moves the distance u over d theta in a time 1 over d theta, but it diffuses a distance square root dx over d theta in a time 1 over d theta. And so if you take the ratio of these two distances, you get a Peckley number, and it looks different from a regular Peckley number because these things together means you don't need a length scale. You have an extra time scale here because of the angular diffusivity. Again, maybe I didn't say that clearly enough, but the angular diffusivity is the fact that the swimmer randomly changes its direction over time following a diffusion. And so this number, this Peckley number, controls how smooth, how straight the swimming is, if you want. So when this Peckley number is small, then the velocity is small and the path looks a lot like Brownian motion. But when the velocity is big, then this path starts looking much smoother. So this is an important Peckley number in the problem. Now, when you study a stochastic process like this, a natural thing to do is to pass to the Fokker-Planck equation, also called the Kolmogorov-Forward equation for this process. And this is an equation for the probability density, the probability of finding a particle in x plus dx and y plus dy and theta plus d theta at time t. So it's the density, the probability density of particle. It integrates to 1 over the domain, at least initially. And so this probability density satisfies the Fokker-Planck equation, and you can basically read off the coefficients from your stochastic process that I just mentioned. So you can just look at the equations here, and there's a formulation to tell you what this diffusion tensor here is and what this angular, well, the angular diffusivity is just the angular diffusivity. So the diffusion tensor is this. Again, it's anisotropic, and the drift, u, the velocity, is u cos theta, u sine theta, right? Because you're rotating a constant velocity in the theta by an angle theta. If you set dx equal to dy, this becomes a diagonal tensor. But in general, we like to study dx not equal to dy because organisms are not spheres, and so they like to diffuse in a different speed depending on which axis of diffusion you're talking about. So there's strong anisotropy in microswimmers. Note here, by the way, that when I write grad, it's the spatial gradient only. So the d theta derivatives are written separately. So again, this is not a new thing. This is a well-known equation. Let me just say a quick note about the boundary conditions for this equation, though. The way you determine what boundary conditions you should apply is you can figure out, you can rewrite this equation in terms of a flux vector. So this is the probability flux, you know, and involves the things that you would expect, the tensor, the diffusion tensor d and theta hat. But if you want to preserve probability at a reflecting part of the wall, in other words, if the organism meets a boundary and you say, well, it's not allowed to leave the system, the boundary condition that you must put at that point is to say that there is no probability flux through the system. So that's the appropriate boundary condition to put on reflecting part of the boundary. So partial v ref here means reflecting, whereas partial v is the whole boundary of your domain v. Okay, so this is the kind of boundary condition that will apply. Okay, so this boundary condition will become important in a second because now I'm going to make something a little bit unusual. And this is work with Hongfei Chen, my graduate student here in Madison. So we have a phase space now which contains the position of the swimmer and its angle of swimming. And people often solve this kind of problem in a infinite space, or if they have a boundary, they treat the swimmer as a kind of point. But what we're going to do here is we're going to give the swimmer some kind of shape. So here, this is kind of arbitrarily picked. This is called the teardrop shaped swimmer. So we pick a nice function that we could write down that was at least a little bit complicated. And so this is the shape of our swimmer. It's, again, it's the teardrop. In this case, I'm thinking of it as swimming to the right in the capital X axis. So, and as it rotates, it's going to change directions like this. So there's a capital X, capital Y frame, which is the swimmer's frame, and there's a little x, little y frame, which is the lab frame. And the swimming direction here is parameterized by this angle phi. So phi is equal to 0 is the swimming direction. So you rotate from this picture to this other picture by using a rotation matrix in 2 dimensions about some chosen center on the organism, which we call the center of rotation. But actually, it's really a consequence of hydrodynamics in the sense it's called the center of hydrodynamic stress, and it plays the role of a center of mass, but for an organism moving in a fluid obeying the laws of Stokes flow. So again, the position of this point can be determined. Basically, it's just in general could be a fairly difficult problem. But once you have it, you have it. This is our new preprint, by the way, just put online a couple of weeks ago. So once you give the swimmer a finite size and you put boundaries, you're faced with a nice situation, or you could call it a problem, but it turns out to be very interesting in that now not all values of x, y, theta are allowed, because some values of x, y, theta correspond to your swimmer entering the solid boundary. So if I give you, for instance, a plane wall at y equals 0, I could write down all of the possible configurations of my swimmer where the swimmer does not enter the wall. So for instance, here you see the swimmer is touching at a cusp, right? And y star of theta is the distance of the center of rotation of the swimmer to the wall. We call this the wall distance function. And as you vary the orientation of the swimmer, this wall distance function changes. And you can study how this wall distance function changes as you roll the swimmer over the boundary. So again, you take this teardrop shaped swimmer, but you roll it on the boundary a bit like a cycloid shape, but the cycloid measures both coordinates, whereas here we're only measuring the y coordinate, the distance to the wall. And so for instance, at a corner point like this, you have to study how it changes from the left tangency to the right tangency point at this corner. And you get a value for this center, this wall distance function, which is, okay, I'll show you pictures in a second. So for instance, let's say you have a swimmer which is shaped like a needle. I will talk about the needle swimmer quite a bit. That'll be my main reference point. So the needle, the picture is here. It's a pretty degenerate swimmer. It just means the swimmer is a segment, right? Just a little segment. Now take that segment and roll it over a flat, a flat boundary. As you roll the segment, the center of the swimmer will move up and down twice, right? Because there's two corners on a segment. So as I roll this little segment from theta equals minus pi to pi, this is pi, by the way, it's horizontal, minus pi is the other way, and pi over 2 is vertical. So the closest approach that a swimmer can take to the wall is when theta is equal to 0 or pi, right? When it's horizontal, it can touch the wall. But when it's at pi by 2, it's the furthest it can be. In fact, it's a half. The length of the swimmer is 1 here. And so this curve here, which again we call the wall distance function y star, is 1/2 L absolute value of sine theta, where L is the length of the swimmer. Okay, so the gray regions are excluded from my simulations because if by the way, I'm not plotting x here because it's kind of ignorable coordinate because the wall is along the x axis. But if I put a point in the white area, that's an allowable configuration, but the gray area are disallowed configurations of my swimmer. So we say that the white area is the configuration space of this system. It is the space of allowable variables, values of the variables. Here it is for an ellipse. So if I take an elliptical shaped swimmer, again, this is just rolling an ellipse over the wall. And you can see that it looks very similar to the needle, except that things are rounded off, as you might expect. And there's a nice formula that you can determine. In this case, there's no corner, so you determine the point of tangency each time. In general, this theory is the theory of convex bodies, actually, because only the convex hull of a swimmer matters as you roll it over the boundary. So you can assume without loss of generality that a swimmer is convex. So it's just a series of smooth curves and corners. Here's the teardrop swimmer. So that needs to be, you can get an analytic formula for the teardrop swimmer, but it's quite nasty. So I didn't write it down here, but you can see that it gets much more complicated. In particular, this dip here is not obvious. That's due to the shape at the front, right? And it's different. An ellipse doesn't have that dip. Whereas the teardrop has a dip. And that tells you something about how a teardrop swimmer would like to orient if it comes near a wall. It tells you that a teardrop swimmer coming near a wall would like to align perpendicular to the wall. It gets stuck in this local minimum at minus pi over 2, which corresponds to the swimmer facing the wall and not being able to get away. Okay. So here's some Brownian simulations of these kinds of micro swimmers enforcing this no flux boundary condition. So the gray area here is what I just showed for the needle. It's asymmetric this time because their center of rotation has been moved from the center. So don't worry about this. It's a small difference. But if I have a swimmer, consider a Brownian path that looks like this. A particle comes near the wall. And again, this is a picture in configuration space. And then it's not allowed to enter the wall. So and there's a drift pushing it down. This is my u sine theta drift in the y direction from the basic Fokker-Planck equation. So there's a drift pulling down here, and there's a drift pulling it up here. So a typical Brownian path will look like that. And if you plot this trajectory in real space, it actually looks like this. It corresponds to the swimmer coming near the wall, aligning with the wall momentarily, and then scattering off when it gets a lucky fluctuation away from the wall. So this is actually not too dissimilar from from actual experiments of swimmers interacting with boundaries. But here the interaction is entirely enforced through the no flux boundary condition to the wall. So we think it's a nice mathematically elegant way of enforcing these kinds of interactions with the walls. What we're missing, the big thing, by the way, I should point out, is that we're not assuming any hydrodynamic interactions of the swimmer and the wall. This is not realistic. We should be putting in these hydrodynamic interactions. Many people have put in hydrodynamic interactions, but we're kind of trying to make a point about the so-called steric interaction, meaning the contact interactions between a swimmer and the wall. So let's look at a situation where we can use this kind of formalism to compute something useful. So consider, sorry for my crude picture, I decided I needed this picture late last night. So this is a swimmer in a channel. So now we have two walls separated by a distance L. And I can ask a natural question, which is if I let this swimmer do Brownian motion as an active particle with swimming in this channel, the channel is wider than the length of the swimmer. So the swimmer will reverse direction once in a while. It will swim, it will probably hit the wall and align with the wall, but eventually it will get a lucky fluctuation that will allow it to reverse direction. So on large scales, the swimmer will start undergoing a random walk. And there's an effective diffusion coefficient associated with that. So what that corresponds to is if you had something like a pipette or something, and you put a bunch of organisms, you would like to know how fast they spread laterally inside of the pipette. Of course, it's a 2D model of this, but we could do a 3D version. It's more complicated, but I mean, let me stick to the 2D picture for now. But I'm going to look for a one-dimensional diffusion coefficient along the longitudinal direction of swimming of the walls. So we have to figure out what the configuration space is. So I just showed you the configuration space for a swimmer a swimmer in one wall. But if you have two walls, you just need to reflect the picture. And again, this bump is because we put the center of rotation not to be at the center of the swimmer. In other words, the swimmer is not rotating. The needle is not rotating about its geometric center. So sorry, this graph is for a needle. If it was for an ellipse, it would be rounded off a little bit at these corners. So again, only the white space is allowed, is accessible to the swimmer. So we want to solve our Fokker-Planck equation inside of this white domain. That would predict the entire evolution of the swimmer. Of course, that's quite difficult, so we have to make a lot of simplifying assumptions. Notice this small gap here. There's a very small gap here, and that's because we've chosen a needle which is length 1.05, whereas the channel has length 1. So this swimmer can barely reverse direction. So making it through that little hole in configuration space corresponds to reversing direction in the channel, because this is pi by 2, it's when the swimmer is vertical. This is minus pi by 2, it's also when the swimmer is vertical. So if the swimmer makes it through this little hole, it means it's reverse directions. So in fact, one way to study this problem is to define a mean exit time to hitting that point. I put a little vertical barrier here, an imaginary wall, and I put a Dirichlet boundary condition of 0 here, and I compute the mean exit time using the exit time equation that I showed before to how long I can expect until the swimmer actually hits that imaginary boundary on average, either this one or this one. Either exit will do. And we call this the mean reversal time. The mean reversal time is the expected time that you can expect to wait before the swimmer goes from, say, oriented with theta equals 0 to theta equals pi, and then back again. Or it'll take another tau rev for it to come back again. And in our paper, we derive a formula for this, which is quite complicated, but it turns out to become much nicer for the special case of a swimmer which has left-right symmetry. So a perfectly symmetric swimmer, like all the swimmers that I've showed you so far. So in that case, you get a beautiful formula for this reversal time, which is unexpectedly simple. Basically, it's the integral of 1 over the probability density. The probability density is the invariant density of the swimmer, is the way the swimmer distributes itself in your channel if you just leave it alone for long time asymptotics. It's basically a steady solution of the Fokker-Planck equation. So what this is saying is that if you find this steady solution as a function of theta, the y variable has been integrated here. I don't want to go into the details, but there is some asymptotic expansion that goes into deriving this result. The point is that the reversal time is the integral of 1 over the density. And a simple explanation for this is that when the density is small, when the probability density is small, it means your swimmer is very unlikely to go there. And that's what dominates this integral. The reversal times becomes large when you have points where the density is low, right? In fact, if the density was 0, it means that you cannot reverse, and this becomes infinite. So in other words, everything is dominated by bottlenecks. And in fact, you can use this to study the reversal time by only looking at the minima of P. And if you do that, you get a very nice expression for the reversal time of the needle swimmer in a channel. This is actually in the limit of fast swimming. So you get a Peclet number, which is not the same as the Peclet number earlier, because there's the channel, the needle length enters and the y diffusivity. So this is a Peclet number for this particular swimmer. And it tells you that, as you make this Peclet number larger, you get exponentially large reversal time. It becomes harder and harder for the swimmer to reverse. And then this allows you to immediately back out an effective diffusivity out of this. Because once you have the reversal time, you can use this random walk picture that I presented, that the needle is going right, but then every tau rev, it goes left again and then goes right again. So every tau rev, there's a reversal, and these are independent events. And so therefore, you can just take the variance of a Bernoulli random variable, right, +1, -1, with this waiting time tau rev, and you get that the effective diffusivity of such a swimmer would be 1/2 tau rev u squared, basically because the step size of the random walk is u tau, and there's so the diffusivity is length squared over time, and the time here is tau rev, but there's a time also in the step size. So you get an expression which looks a lot like what people use for turbulent diffusivity or something like that, right? When people write an eddy turnover time or something like that, they write a tau here, but here it's a swimmer turnaround time rather than an eddy turnover time. And this is actually, we show it's actually rigorous in the sense that this is actually an upper bound to the effective diffusivity. It's not just an approximation. We can also use homogenization theory to capture a much more precise result about the effective diffusivity. In other words, this is a very nice, I think, heuristic formula based on the reversal time, or a bound, and a rigorous bound based on reversal time. But in our paper, we also present a full-on calculation of the effective diffusivity, which gives corrections to this, basically. Okay.
Question.
Yeah.
Professor, is d theta equal 1 in those previous?
Oh, yes, sorry. The time scale is d theta. Yeah. So yeah, that's exactly right. That's like saying d theta is equal 1. Yeah. So d theta inverse is the time scale. Thank you, Charlie. Okay. So let me conclude. There's a long tradition of studying stirring and mixing at the GFD program. And, as I pointed out, even when the summer is not on stirring and mixing has that ubiquitous presence in all sorts of disciplines, where there's very few people who consider it their field of study, but everybody needs it, right? So I would say that it's been studied in many, many summers at GFD in one form or another. And in fact, some important innovations were pioneered in the program, as I discussed. And in particular, what I talked about at the end of my talk here, I say transport and mixing of microswimmers and due to microswimmers are both interesting questions, right? So there's the biomixing question about how swimmers actually stir their environment, and it can be relevant in some biological situations, but also the transport of the microswimmers themselves, which is what I talked about in the last part of the talk, which is just treating the swimmers themselves as particles and watching how they get transported around. So this is still a very active area of study. And of course, you can get lots of anomalous behavior and all sorts of exciting things. In general, the interaction of microswimmers with boundaries is a massive topic. And I really apologize here. I didn't really do a justice to the literature. It's really quite a large literature. I point you to our paper where we really dug through the whole literature. And there's many, many ways of approaching the problem, a lot of it numerical, in fact. Our focus in my current work that I discussed at the end with Hongfei and several others is this idea of using configuration space and actually solving partial differential equations in configuration space to really capture the shape of the swimmer as a parameter and varying that. And so in our paper, we look at ellipses and segments and things like that. But you could even think having deformable boundaries. You can have a swimmer which actually changes its shape, and the configuration space picture is still valid, except now it's a problem with a moving boundary, right? But in principle, there's no obstacle to actually solving these problems. So again, the idea of configuration space is that the degrees of freedom to swimmers are limited by boundaries. I'm not trying to say this is a new idea, by the way, right? But we haven't seen it applied to microswimmers very much. And as I said, we're missing a lot of things that we're not putting in. And I think the biggest one would be hydrodynamics, right? We're not putting the swimmer and the boundary do not modify the flow. There is no flow to some degree in this model. The only hydrodynamic inputs are the drag, the way you compute the diffusion coefficient of the swimmer come from Stokes flow, fluctuation dissipation theorems. But there's no other hydrodynamics in the model that I presented, which is a virtue and a flaw, and it makes the model simpler. And I just want to apologize at the end because there's many fellows that I work with over the years that I did not mention in this talk, Anshuman, Michael, Kiori, Amanda, and Tiffany. But I couldn't fit everything into the time allotted. Okay, thank you very much.